Hello, Professor Greg, and thank you very much for joining us here. I am Kıvanç, final year PhD student in NTU MSc. So we would like to start you with, how did you first get the idea to use phages to evolve antibodies?
Well, it kind of followed on like most ideas. We'd had this idea of creating large antibody libraries by shuffling the heavy and light chain genes. We would need to screen the libraries for very rare antigen binders. George Smith had published a method called phage display, in which he'd used to isolate peptides on phage. Now, we thought maybe we could use that same method for antibodies. But actually, the big unknown was whether we could actually display antibodies on the phage. Turns out several people had the idea independently from us at about the same time, but it's actually technically very challenging. There are lots of reasons why it might not have worked. So that includes the fact that it was well known that M13 phages, if you put I'd worked with DNA sequencing, if you put a large insert in, as we'd have to with antibodies, they tended to delete out. It was also the case that we were putting a protein into the gene 3 at a point where this protein mediates the infectivity of the phage. So actually we had every reason to believe that it might abrogate the infectivity of the phage. It also wasn't clear to us whether the fusion protein we'd make would actually fold up properly. It also wasn't clear whether the antibody might be cleaved off, because there's various sequences around it. And quite often you can try to make a fusion protein, you find it's cleaved. And there's the further issue. If you look at phage, look at whether it binds to a solid support, it tends to be sticky and tends to bind to solid support. So we had to deal with all of those, some things which didn't turn out to be an issue and some things which certainly were an issue. But we anticipated them and we took the precautions and we persisted and we got it to work.
Thank you very much. And the second question?
Your foundational work in antibody humanization paved the way for blockbuster drugs like Herceptin, Avastin, and Humira. How do you feel seeing your early innovations translate into real-world impacts?
Well, obviously, I was pleased and actually somewhat surprised that both the science and the translation worked out so well. And I have to say, it was touch and go on the translation. I think that could very well have gone in a very different direction, and we were very lucky that it worked out as it was.
I'm Kestrel Tang. I am a Year 3 NTU student, undergraduate from the Faculty of Chemistry. So I'm quite curious because a lot of our viewers may not be specialized in your field of research or biology for that matter. So could you perhaps explain your Nobel winning work in the way you would explain to someone who has no prior experience and understanding of your work?
Yeah, well, that's actually a very difficult question. And thank you for letting me know about it in advance, because it's taken me a lot of time trying to think how I would explain it to such a person. But let's put it this way. If you need to open a lock, but you haven't got a key, what do you do? Well, one answer is you can take a large bunch of different keys and try each of them in turn until you find one that fits. And if the bunch of keys is big enough, it would be possible to open any lock. Now, in the world of molecules, we're faced with similar problems. We might need to make an antibody that sticks to a cancer cell and stops it dividing. So what we do is to create billions of different antibodies and see which ones stick. And my Nobel Prize-winning work showed how to do this. And this is the really tricky bit that I'm struggling to explain to someone with no background. But essentially, we created billions of different antibody genes, and we inserted these into a virus so that the virus displays that antibody fixed to its coat. We then added the viruses to the cancer cells and washed away all those that don't stick. And we then took the cells with the viruses attached, we isolated the antibody genes from them and built these into complete antibodies. But I can't simplify that work any more than that, really.
Thank you very much. So the next question would be is that one of the biggest advantages of phage display is that it doesn't inherently require the use of mice or any animal testing during the core antibody discovery and selection process, but rather it's a completely in vitro technique. What lessons can we learn from phage display to discourage the use of mice testing in therapeutics?
Okay, so with phage display, we're essentially mimicking the way the immune system makes antibodies. So perhaps the biggest lesson is that it's sometimes possible to avoid the use of animals in some parts of the process for making therapeutics by mimicking some natural process. But actually, the main use of animals in development of therapeutics is driven by regulatory requirements. It's not driven by the discovery process. Discovery we can often get by without using animals, certainly with phage display, but the real requirements come when you've got to decide you want to put a drug in a human, and frankly, probably, I would rather see that go into an animal than, for example, go into my child as the first, or into me as a possibility. So I think that's really where the bulk of animals get used in preclinical testing.
Then what do you think are the limits of phage display, and are there things it can't help with?
Well, it's certainly true. Time has shown that it isn't an answer to everything. I mean, ideally we need folded, preferably pure proteins that can be coupled to a solid phase. It's actually quite difficult to succeed with phage display if the antigen is a small part of a mixture of proteins. It's also difficult to use with membrane proteins, and that's because it tends to stick to the membranes. And it's also difficult to make phages against specific carbohydrates, to make phage antibodies against specific carbohydrates. Because the problem is carbohydrates only bind weakly to proteins. That probably reflects the fact that you've got a lot of hydrogen bonds, and for hydrogen bonds to be strong, they've actually got to be they've got a very preferred orientation. Electrostatic bonds they simply go as a power of the distance between them. But hydrogen bonds require a distance and an angle, and that means it's quite difficult to get a high affinity hydrogen bond, and of course sugars are full of hydrogen bonders. If you look at the sugar binding proteins, most of those are multimeric. They've got multiple domains, or they've got multiple motifs within the same domain. And it is that which allows you to get the high affinities. Antibodies, we've just got a single binding site. And so unless we express them in a particularly multimeric form, it would probably be difficult to expect to get a high affinity of a single antibody head to a carbohydrate.
Greg, yes, I was fortunate enough and lucky enough to listen to your presentation yesterday, and I realized that you've really come a long way in your research. So, when you were researching on phage display and coming out with it, was there any moment in time where you realized that perhaps it wouldn't work and you almost gave up on it?
Not really with the phage display. I mean, because we anticipated problems at the beginning, we managed to step around them. We had the right reagents. We had, for example, antibodies that would identify whether the phage was sticking to a surface. It so happened I had a freezer of goat anti-phage antibodies that Fred Sanger had given me 10 years earlier when he'd finished working, when he'd retired. And I didn't know what I'd use them for, but I thought that might come in useful one day, and I'd put it at the back of my freezer. And that turned out to be absolutely a key aspect of checking on the extent to which we could block the non-specific binding of phage to surfaces. But we put in place the techniques and controlled, and actually it all worked first time. And in fact, we kept being surprised at how well it worked, because all the things we'd anticipated, we either dealt with them or it didn't turn out to be a problem. I mean, for example, we were worried about the phage deleting the genes. And certainly if you put a big insert in an M13 sequencing phage, it will delete with high frequency. And we were putting big inserts in. So why don't they delete? Well, in fact, they do delete. But if you've got, what of course we hadn't got with the M13 phage was we didn't have a selection pressure on maintaining the insert in place. What we had with the phage, we'd got an antibody on the tip of the phage. So the deletions, essentially, we didn't see. We only got the ones with the antibodies that were actually binding. Now, the deletion issue does become a problem if your selection isn't very efficient. because then, in fact, the frequency of deletions can overwhelm the population. So if the frequency effectively is very low, that's a problem. And if the selection isn't efficient. But in general, it didn't turn out to be the problem we thought it was.
Thank you. So apart from the technical questions, we also would like to hear you about some personal questions. And here the first question of us is that what was the biggest conceptual hurdle you had to overcome, and how did it shape your thinking?
Well, going back to phage display, there was no conceptual hurdle. Since once we had the idea of making large antibody libraries, it was a kind of phage was one possibility for trying to screen those libraries of many. One possibility would be mammalian display, yeast display. So we reckoned we probably got to try all of them before we'd find one that would work. But more generally, I focus my work in areas where I don't have conceptual hurdles. In particular, I'm happiest thinking about molecules and their structures and trying to explain biology in terms of those interactions. The area I did actually struggle with conceptually, certainly in the 1970s, was immunology. I could not understand what they meant by MHC restriction. I couldn't really understand what they meant by tolerance. And it took an understanding, it took the further development of that field to understand those in terms of structures that meant I began to understand immunology as a whole, because I couldn't understand the concepts as they were put in the form when we couldn't think of it in terms of molecules.
One thing researchers go through is that there's always a repeated failure in experimentation, and it kind of weighs many researchers and students down. So what advice would you give to these students and researchers when they are faced with such hardships and failures?
Well, I kind of saw my career path as akin to a sort of medieval apprentice, working with a master to acquire skills and a trade. And so I focused on learning those technical skills. I kept experiments simple. I included sufficient controls. Actually, I took heart from some advice from Fred Sanger, who, of course, was one of my mentors and a double Nobel laureate. And I remember going and pointing out that this experiment wasn't working. And he said, well, should it work? And I said, well, I think it should. He said, well, if an experiment should work and it doesn't, you usually just have to fiddle with the conditions. And that has been one of the most important things I learned in my work. Usually, you've just got to play variations, and then if it really should work, then it usually will.
All right, so Greg, as you know, you've been honorarily appointed as the IAS Lee Kong Chian Distinguished Professor. A two-part question for you. So how do you think that resonates with the broader engagement and global scientific academic community? And more importantly, I think as you have been speaking at the IAS 20th Anniversary Symposium yesterday, how do you think is the importance of engaging younger institutions like NTU through this kind of academic exchanges and visits?
Well, I suppose I've had a fulfilling and exciting career in science, and I do consider engagements with institutions or individuals, irrespective of their age or any formal appointment. In fact, usually appointments and contracts have usually come with some kind of expectations or obligations or even legal responsibilities, and to the extent possible, I prefer to avoid those. But I do feel a duty to engage with younger scientists, and older ones as well, actually. But there's not many of those left. And to tell them just how great science is and its applications can be. But I'm kind of now retired, and I don't really think about formal academic exchanges. I used to have to think about those when I was, in charge of an institution, but now I'm in charge of myself. So I have a different approach to things.
And thank you for your response. And this question is my personal question for you that after I read some information on you and after I see most of the rewards that you got. So my question is, what inspired your early interest in science? And did you have any role models or mentors growing up?
If I look back, there are a couple of events that did inspire me to do science. And I was probably about 5 years old. I was brought up in West Africa, At that stage, it was a British colony. It was a colony of the Gold Coast, country that later became Ghana. And I was, my education started in a small mission school set in a clearing in a bit of the savannah and near a forest. But it was close to, my father worked at the University of Legon in Ghana, or in the Gold Coast, then Ghana. And the university academics would sometimes come at our break time and they would show us things. And I can remember at that time, there was we had a visit from a scientist from there who was looking at radioactivity. Now, at that time, the French were exploding, I think, atom bombs in the Sahara, and there was a lot of concern about the radioactivity blowing from the Sahara, which it did on those winds then, into the Gold Coast. And he was also particularly concerned, and he was monitoring radiation levels. And he came with a Geiger counter, and we had great fun going around the school trying to identify things that were radioactive. And what the most delicious thing I remember was he put it at the very end. We were all a bit disappointed, we hadn't found anything terribly radioactive. Then he called across the teacher and he put it on her watch. Now, in those days, they used to paint the hands of watches with radium, and so they would be illuminated in the dark. You could see them illuminated. And this Geiger counter went off scale. And that teacher was so horrified, she stripped her watch off immediately. She was going to talk to her husband about that because it's been a present from him. And it was the combination of the curiosity and the unexpected that caught, I think, just caught our imagination. We thought it was wonderful making this discovery. Then, probably a few months after that, we had a visit from a biologist who came with a large tank, and we could see there was something writhing about in the tank. And we looked over the edge, and in fact, it turned out to be a large turtle. And I'd never seen a turtle before. And some of the African workers there were frightened that they told us to get back because it could cut our fingers off and it might leap out and jump on us, which seemed pretty unlikely, I have to say. But anyway, this scientist, it turned out, said, well, don't worry, don't get too close, but don't worry about it too much. And I asked him what he was, because I thought this was very thrilling to have a job where you brought in wild animals. And he said, well, I'm a scientist. I go out, a marine biologist. I go out into the sea and we capture wild things and we bring them back. I said, well, what do you do? He said, well, then we experiment on them. And now I hate to imagine what happened to the turtle, but I thought that's what I would like to do. I'd like to go into the wild unknown and capture wild animals and then experiment on them. And I didn't know what it meant, but actually, to me, that was a career already mapped out. So that definitely had a big effect on me. Then later on, that sort of set me on the right path, and I started thinking about how I would do experiments. And I remember experimenting with a chemistry set I was given. I remember my brother and I, with this chemistry set, we had some hydrogen peroxide, and if you dropped a bit of potassium permanganate crystals into it, you'd get oxygen being released. So I do remember we caught a wasp and we put it in a test tube and then we fed it oxygen. And the buzzing went absolutely intense. Anyway, we tried buzz testing wasps to destruction to see how long they lasted. It's very, very interesting. And then to dampen it down, we fed it carbon dioxide, which we got from somewhere else. And then usually we released them. I think there were very few that we destroyed. But usually we damped them down with carbon dioxide, ran to the other end of the garden and hurled it out, and then ran as fast as we could in the opposite direction. And so again, it was learning, it was trying out. This was a world with animals and insects and things like that, and the freedom to do those kind of experiments. So I think I was hooked really from primary school level and then various stages throughout my my career, and I've always wanted to be a scientist.
Thank you for sharing some of your personal life information. So now we will move on to the next section, which will be questions about the future of this field. So how do you see protein engineering evolving over the next 20 years, especially with AI-driven tools like AlphaFold?
I think we're going to see the further application of these tools, in particular to tune the protein affinities or activities or selectivities. It's going to require a correlation of sequence, structure, and biophysical data. Now, what we're missing at the moment, although we've got rapid methods for getting sequence and to some extent structure, we're missing a lot of biophysical data, hard data like rate constants and equilibrium constants. And I think what we can expect to see is that being automated much more, and the collation of huge databases in which sequence, structure, and biophysical data are correlated. Once you have that information, I think that will lead to greater ease of generating proteins with required functions, and may even include generating fairly automatically. So that's what I think we'll see.
So, I mean, as you know, AI and artificial intelligence tools have been sort of the hot topic in recent years, and especially when it comes to the STEM field. Do you think AI would eventually replace techniques like phage display?
Eventually, one day. But we're not there yet, at least for therapeutic antibodies. And again, that's not in the big picture, designing protein is only part of the story. Developing a therapeutic is another story.
Thank you. So we also wonder what excites you most about the future of biological drug development?
There's a huge range of platforms already for making drugs, from small molecules, peptides, proteins, antibodies, DNA, and RNA. Currently, the excitement in the future will come from seeing how those things get applied and the diseases they can cure. There's perhaps one area that's underdeveloped, which I hope gets developed, which is the use of cells for therapy. I mean, that's got huge potential and also huge difficulties.
Then how would you envision the future of cancer treatments evolving with the combination of phage display technology and advances in AI?
Well, we can already make antibodies for treatment of cancer, whether by immunization or by phage display, perhaps even by AI in the future. But the problem is finding the right target for the disease in question and undertaking the clinical trials. And actually, I think it's those areas that AI has got the most to offer. It's not really an antibody discovery, because that's not rate limiting. The other areas are.
Greg, given that you mentioned earlier that your scientific journey was sort of like a medieval apprenticeship. Given that, what skills or mindset do you think young scientists today need the most?
Well, so moving on from the mindset, I think you need, modesty prevents me saying this really, but you need curiosity, intelligence, hard work, practical skills, and a passion for the subject. One of the things that most impressed me when I first went to work at the Laboratory of Molecular Biology is to hear scientists, in particular Cesar Milstein, arguing with my own supervisor about scientific questions with a passion that was almost like politics, like you'd hear people arguing about politics. I was astonished at how wonderful it was. People could get worked up about exactly how a protein will cross a membrane and the competing theories about it. And I think that's good. I think one should be passionate about a subject. I would say something else a young scientist needs to do is work things out for themselves rather than just look at the answer from the web or from AI. I mean, even if AI and the web gives you a better answer, I think actually it's better to try at the beginning to work it out yourself, because that enables you to develop your critical thinking.
Thank you. So as a follow-up question, we would like to ask, what advice would you give to young scientists aiming to translate their research into practical applications?
Well, I would say first, develop your research and technical skills, preferably in multiple areas, so you're not just a one-trick pony. Focus on important questions, not just interesting questions, but important questions. And if you see a good opportunity for translation, seize it, not forgetting to take out patents on your inventions. And because that combination of your skills and intellectual property will be required if you want to persuade anybody to give you money, or you want to persuade anybody to license your technology. Bear in mind that you'll have to combine your scientific skills with management, commercial, and legal skills. And you'll have to learn to work as a team rather than a loner.
As we all know, phage display has revolutionized the biologic drug development. So what do you think are some similarly hidden exceptional technologies today that are waiting to reshape another field, perhaps outside of biology?
Well, within biology, I've already mentioned cell therapy, I think is an important area. I also think this is kind of on the borderline of biology and chemistry, is that recoded organisms that can build new proteins with novel amino acids will have an impact in biology and probably in material science as well.
As we know, you have visited many universities and institutions all over the world to share about your research and developments. So, as someone who has worked, whose work spans academia, biotech, and translational medicine, how do you think these universities, or what do you think these universities could do to better support innovation and entrepreneurship?
Okay, so I think universities could offer free, short business courses to all scientists to include patents, licensing strategy, setting up and managing a small company. I also think they could help to forge links between their business-savvy alumni and scientists, not with themselves always standing in the middle, being a mediator, but actually allowing direct links. I also think universities should give scientists more freedom. I mean, how do you ever create a cadre of scientific entrepreneurs unless you give scientists a free hand in exploiting their inventions? What we don't need is more bureaucracy. And there's a tendency of universities to say, yes, we'll support it, which means putting more money in and more bureaucrats to mastermind the process. In my view, a bureaucrat never masterminded anything. They just get in the way.
So in the previous questions, you briefly mentioned that the researchers should pay attention to the teamwork and collaboration. In this regard, how do you see the role of collaboration evolving, both across disciplines and across borders, in a world that is sometimes increasingly fragmented?
Well, look, how it evolves is going to depend on the scientific field, on the local ecosystem, on the finances available, and lots of other factors. So, for example, collaborations in research with military applications have already been enhanced between some countries and inhibited between others. Conversely, another pandemic might actually see better collaborations between all countries. So I think it's immensely dependent on how the future evolves. But what is unquestionably the case is there are great advantages in collaboration if we can do it.
Many of today's biggest breakthroughs happen at the intersection of disciplines. Are there any scientific fields outside of your own that you have personally found particularly exciting or inspiring recently?
Well, I would say the idea of encoded organisms, perhaps interfaces between organisms and electronics, including but not limited to the Musk Neuralink project in which it's proposed to link human brains with computers. Those would be areas that I personally find very exciting. An obvious area that has excited me for a long time has been the origin of life. In fact, it got me started really in science at university level. And the idea of how organisms first developed and how they evolved, particularly in the pre-Darwinian phase. But I don't think I'll ever see the answer to that in my lifetime.
Thanks, Greg. I think you've come a really long way in your scientific journey. So looking back, if you could speak to your younger self at the start of your scientific journey, what would you tell yourself? What advices would you give yourself?
I think I just say carry on.
Short and simple.
Okay, thank you. So we would like to finalize with this question. What do you hope your scientific legacy will be, not just in terms of discoveries, but values?
Well, I hope scientists will be inspired to do exciting and important work and to engage with the world as it is to make things better for all, including themselves.
Well, if there aren't no further questions, Greg, do you have any last anecdotes to share with the entire panel here? Just off the cuff.
Off the cuff, no.
If not,
I have to try to think. If I had to think, I would say, I think, 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 I think
very, very hard.
Or perhaps for young scholars emerging in the field, just starting their careers, figuring out what they really want to do, whether they should enter industry or whether to just stay with academia, or what do you? I know you possibly handled this question slightly earlier, but what else do you think they should develop about themselves as they move through this new world?
People are too concerned with trying to identify what they're going to work on for the rest of their lives. My feeling was that you just choose something, you make a choice, and then opportunities, if you immerse yourself fully in the field, opportunities will emerge. Or they won't. And if they don't, then you find another field in which you go into. The worst thing to do is to sit around agonizing. If you're uncertain, you're really uncertain, you're doing the wrong thing. Perhaps you shouldn't be in science.
All right, well said. Thank you very much, Greg. And I guess on behalf of the students and staff of NTU, thank you very much for your time and traveling all the way to Singapore to answer our burning questions, which we really wanted to ask you for the longest period.
Well, thank you for the questions. Actually, they've been quite interesting to think about and to answer. So thanks.
Thanks, Greg.